Resources in the world are typically scarce. Animals typically search for resources, and to be successful at this search, one could utilize special search strategies. Many studies have been addressing if some search strategies are better than others. For example, in terms of time that a searcher spends on finding a resource. T lymphocytes are cells of the adaptive immune system of mammals such as mice and humans. They are tasked with the job of locating and eliminating cancerous cells or cells infected with pathogens such as viruses or intracellular parasites. How exactly T cells accomplish this task remains poorly understood. Do T cells randomly move around in the body until they stumble upon their target? Or do they employ more sophisticated search strategies, for example, with some local search and then long displacement to another location?
T cells are now an important part of novel strategies of cancer treatment in patients. These methods either stimulate T cells in the body to attack cancer cells, or T cells grown in culture are transferred into patients in the hope that these T cells eliminate cancer. Better understanding of how T cells move in the tissues to find their targets and how we can perhaps regulate these movement patterns may make cancer immunotherapies more effective.
So what are the movement strategies of T cells? For a while, it was not fully clear. Some studies suggested T cells perform random walks like the movements of small particles suspended in a solution called Brownian walkers. Such movement pattern typically allows thorough exploration of local areas, but Brownian walkers typically do not displace far from the starting place, often returning to the position they started from. A pioneering study by researchers from the University of Pennsylvania used microscopy to image movement of T cells in brains of mice infected with the parasite Toxoplasma gondii. T. gondii is a eukaryotic parasite typically transmitted by cats to humans and may be very dangerous to the developing fetus in pregnant women. By careful tracking positions of the T cells and by analysis of the movement data, the authors proposed that T cells in the brain perform generalized Levy walks. In these walks, T cells pause for random periods of time characterized by infinite variance and then move in a random direction for times also characterized by infinite variance. The authors proposed that such a movement strategy may allow T cells to efficiently locate and eliminate rare parasite-infected cells in a complex tissue such as the brain.
One of the key issues with imaging data on movement of T cells in tissues is that in a typical experiment, T cells can be readily detected, but other cells and environmental constraints of that tissue are typically not. To paraphrase a statement by Ron Germain, a National Institute of Health scientist, the eye is biased by the things it sees and not by the things that are invisible to it. Because we don't know specific constraints in the brain that may bias movement in brain-localized T cells, attributing the specific movement program to T cells may be premature. We have recently performed experiments in which we followed movements of T cells localized to the liver. Interestingly, we found that the liver-localized T cells exhibit various movements like pausing, directed movement, or rapid displacements. We developed a methodology to rigorously image and digitize liver sinusoids, which are the blood vessels of the liver used by T cells to explore the liver environment. By taking the structure of sinusoids into account, we could actually explain the T cell movement program by environmental details. For example, straightness of the sinusoids forces T cells to move in nearly straight lines, and the blood flow pushes these T cells to displace rapidly and far. Such rapid and long displacements are the cardinal features of the Levy walks. We therefore wondered if perhaps unique features of the brain may explain why brain-localized T cells perform generalized Levy walks.
Luckily for us, researchers in Australia recently performed experiments where they imaged brain-localized T cells after malaria infection and shared these data with us. We have used our recently acquired software called Amaris to accurately map coordinates of T cells in 5 separate data sets. As we analyzed the data, we discovered that T cell movements were not uniform, and in rare cases, T cells appear to displace far. This is a feature of Levy walks. A closer look at such movements, however, showed that they arose due to artifacts and digitization of cell positions. After cleaning the data, we found that brain-localized T cells search for the infection using correlated random walks, a type of Brownian walks, and not generalized Levy walks. This suggests that brain-localized T cells search for an infection site randomly by performing mostly short displacements.
Our results suggest that T cells in the brain use a relatively simple movement program to search for infection, short movements in one direction and slow change in that direction over time. Such correlated random walks may still be sufficiently efficient to help survey larger brain areas. This puts a strong limit on the brain volume explored by these T cells to locate and eliminate all the infected cells in the area. Our work thus provides a limit on how many T cells an efficient vaccine must induce in the brain in order to eliminate the infection from the whole brain.
Results of the work of the scientists from the University of Tennessee suggest that brain-localized T cells use simple movement programs to explore the tissue for infection. Knowing the movement program, the number of T cells, and their speeds would allow us to predict the time T cells would need to survey and eliminate infections or cancer in the brain. These results naturally lead to a framework to evaluate efficacy of T cell-based vaccines. Vaccines that induce insufficient numbers of T cells to provide protection will then receive a no-go recommendation for large Phase 2 or 3 clinical trials.